Hi, everybody. Thanks for coming today. So my name is Tiffany. I also go by TJ, which you see on the syllabus. I did my PhD in the Nolan Lab, and I graduated about a little more than a year ago. And now I'm at a company. So my research primarily focused on cell cycle and drug discovery. So if anybody wants to talk about that later during the course and things, that's a personal interest of mine. Also here today is Nikesh in the back. Wave to everybody. So Nikesh founded Cytobank, as well as with Peter and Jonathan, who I'll mention briefly in this talk. So Cytobank, for those, how many of you guys have heard of us before? Oh, nice. So Cytobank is a platform for managing, sharing and storing your cytometry data. And this is a company that was homegrown out of the Nolan Lab actually I think it's the 5th anniversary now, pretty much, no, more than that. I think it's the 5th anniversary of some of your first hires, I guess. And so we're actually located nearby here in Mountain View. And I'm actually here in the Nolan Lab quite a bit, working with Wendy over there. So the general idea of Cytobank is that you can manage, analyze, and share your cytometry data over the web. When you log in, and you can sign up for a free account online, you manage your data using the inbox, which is where you look at all your different experiments and store that information. I'll also show you all a bit today about how you can store all the metadata, as well as all the way down through the analysis and the figures. Cytobank was founded by Jonathan, Nikesh, and Peter, all from here at Stanford. And they have a number of patents as well as publications in the cytometry field. You can get to us in 3 different ways. The first way is through the freely available version, which is cytobank.org. I highly recommend that if you have time today to sign up on dvs.cytobank.org. Tomorrow, you'll be going through a spade demo that Rob Brugner is going to show you from the lab, and he's going to be using DVS. In addition, you will analyze the data from the course in Cytobank. And so you might do a little bit of that upload today, and then you'll be doing some analysis tomorrow afternoon as well, using some of using SPADE as well as some of the other premium functionality, which is available on DVS Cytobank. DVS Cytobank is on a per license basis, and so you can sign up and it's a free trial. You can get going and started individually. And you have access to things like SPADE, dose response analysis, and plate annotation, which you don't have available to you on the free community version. The third available version is Enterprise Cytobank. For example, Harvard, UCSF, Stanford, they all have their own cytobanks. This is for either at the lab level, for example, Nolan Lab has their own, or at the university level, where a lot of people can come and have an internal place to share and manage their data together. Independent of that, we have a large education effort where we're trying to give people examples of ways to analyze their data. For example, we have example dose response analyses, SPADE plots. How many of you have heard of SPADE before? Oh, not that many. OK, cool. I'll talk a little bit about it as we move forward. Gating strategies, because as many of you are going through right now, you're not only learning phosphoflow, but you're learning mass cytometry and kind of the way to go through and analyze that data, because you're new to a lot of these different techniques, as well as heat maps, histograms, and contour plots. In addition, if you're interested in finding out more about us, we have our cytobank.inc website, which includes kind of news about the company, other resources, a lot of helpful resources actually, and also job opportunities, because we are growing right now. So I just wanted to kind of talk a little bit about some of the new motivations for the company and how we've changed over the last couple of years. So one of these big things is that high-dimensional cytometry has really pushed forward how we do data analysis, how we do our experiments. So Cytobank is a place where you can analyze your data, but also view many different types of plots. But really, as we push towards high-dimensional data, we actually host a number of papers on Cytobank itself. So you can actually go and access the raw data as well as the figures associated with them. For example, I'll talk about a little bit about these more later, but there's the original science paper, but there's also, for example, the cisplatin dye. There's the original paper for that. There's cell cycle information. Greg's giving a talk on that tomorrow morning, as well as drug discovery and Rachel's normalization tool, which is also hosted on Cytobank as well. So Cytobank was actually founded on caliber data, so 4-color data, but it was at the point where there was just too many samples, and it was really hard to think about and work with that information. And so really, if you go and you can put your fluorescence data on Cytobank, you can put your mass cytometry data on Cytobank. But we're addressing a lot of the needs down here in kind of the low parameter and kind of low number of sample space. But really, we're pushing towards the future of flow, which includes high dimensionality as well as high parameter analysis, and really the communication and collaboration which comes with that. From the person who designs the experiment to the person who's like potentially the technician working through it, as well as to the clinician and the researchers making those actionable decisions at the end. And this really is a team effort, if you think about it. So this is an example set of 2D plots from the original science data set. Now, you know you wouldn't actually try to go and try to gate through all of these populations individually. Some of them don't necessarily make sense. But you could imagine that looking at one section, you might be thinking about, well, this section down here are my T cells. That section up there are my B cells. And if I'm purely a T cell biologist, then maybe I'm missing something, right? Some kind of interaction between the data, some kind of interaction between my cell subsets within the depth of my experiment. And the idea is to kind of take something that's just huge like this and reduce it to the results that you're trying to find. And really, as we move forward, because of kind of the high dimensionality of mass cytometry, really data analysis is becoming the major bottleneck. And people are coming together in whole teams, sorry, moving forward and analyzing this type of data. In addition to the depth of this information, there's also the breadth. So there's a paper that we published in 2012. So Bernd Modemiller, who was a postdoc in the lab and who's now in Switzerland with his own lab, published this experiment wherein basically one tube barcoded 8 inhibitor concentrations, 12 stimuli, 14 signaling molecules, and gated 14 populations to end up with over 18,000 measured conditions. And so we're getting to the point where with one CyTOF run, you can put this much information. And so you were thinking about it, what you're going to do tomorrow and today is you're going kind of in this depth of information with your signaling studies, with the number of markers that you're measuring, but really the possibility with using barcoding and other techniques, you're going through that breadth as well. And that's why we designed Cytobank, not only to address that depth, but that breadth, and to really just simplify the ability to work together in teams for the analysis of these larger experiments. And at the very core, preserving and managing this data is key. So this plot right here on the left, so 90% of all data ever generated has been generated in the last 2 years. Okay, so the first time I saw this statistic, I kind of laughed at it because I was like, how do you know that 90% of data has been generated over the last 2 years? Well, kind of narrowing that down a little bit, scientific data is growing at a rate of 30% per year. And that's a figure that I will definitely trust when I hear that. Because if you think about, when I started my PhD and how I was collecting maybe 4 or 5 samples, and now what you'll see is some of the data that I've put in Cytobank, I'm collecting plates and plates, 90 different conditions, for example. And the rate at which scientific data is increasing is huge. But the huge issue that's stemming from that is that we're losing all of this information. A paper published in this last year by Vines, they noticed that retrieving any data from publications is decreasing by 17% per year. And this is based on the age of the paper. So the x-axis here is the age of the paper in years, and the y-axis is the probability that you can't retrieve the data anymore. And it's not just a linear slope. It's actually kind of dipping down extremely quickly. The second issue is that results are not reproducible. A 2009 paper, which primarily focused on microarray data. If you think about microarray data, it's increased a ton, right, in about 2001 to 2011. greater than 50% of non-reproducible results were not due to bad science or anything like that. It was due to the fact that people couldn't find the data and they couldn't find the software. And so this is a huge problem that we're trying to solve as a company. And so if you think about it and how it affects us, right, currently in terms of us as researchers, what do we need in terms of the single-cell cytometry research? And one of them is just an easy way to back up and share and store your FCS files. The second is to share with collaborators and work together across departments or geographies. If you think about any pharmaceutical company or any collaboration that you might have, no one is working at just one site anymore. There are multiple sites, and a lot of, if I think about the pharmaceutical companies, they're trying to consolidate all their databases right now. For example, Stanford's initiative right now is to consolidate all of their clinical data as well as a lot of their kind of patient data from the single-cell biomarker and single biomarker databases. In addition, a platform for large computations and novel visualizations. One of the things that's very difficult but also wonderful about flow cytometry data is the fact that you can easily collect millions of cells, which is a statistician's dream. However, to actually compute and cluster and do analysis on that is quite difficult to do on just my laptop alone. In addition, so managers and PIs, a big concern is about losing your data when people leave the lab. Ron Levy and Gary Nolan here, they were the first people to get their own cytobanks. And Ron Levy has data from 10 years ago or more on his cytobank. And I know if I think about data that I tried to collect 10 years ago, I have no idea where it is right now. I don't know if you can think about that right now, but yeah, I was doing butterfly research 10 years ago, and so I have no idea where that data is now. The final thing, which I think is really the foundation of Cytobank and where we really support researchers, is the fact that you have your experiment details linked all the way down to the analysis. And what this means is that you have a gradient in terms of the granularity of your research. For example, when you collect your data, it's down here at the raw FCS file level, but you might do compensation. You might transform your data, and then you will go ahead and gate through. And then at the highest level, you'll see your heat maps, your histograms, your 2D plots, and then you'll have these, you'll export them and put them into PowerPoint figures, right? And the idea is to put that all in one place where it's all layered so that if you tweak, your compensation, if you tweak your transformation, your figures will reflect that. Your heat maps will change if you change your gates, and that will stay all in one place. And so that really helps the reproducibility of research, and as you share with your collaborators moving forward. And what's really the beauty of this is because we are a web platform, because you can go and analyze your data online, you really think about your results, and you don't think necessarily about the infrastructure behind it. So spate analysis is a powerful tool for analyzing cytoph data as well as fluorescence flow cytometry data. And when I started in the lab, I was running my analysis for cytoph data on a compute cluster, which was basically, I think, like 80 of my laptops, I guess. And that would take about 20 hours to run. And so now what you can do is you can actually start an analysis on Cytobank. Actually, you can do it from your phone, even if you want. And then what you can do is you'll just close your window. You'll go to bed. It'll email you when it's done, and you can check it afterwards, and you can actually go and look through your analysis. Okay. So sorry, I hope that didn't sound too sales pitchy, but I do love the company. And so really, having everything in one place is really crucial to working together in a team. If I think about the teams that I've worked on, typically they're anywhere between 2 to 8 to 12 different people working on a project together. And so how does it actually look? Like, how do you actually work with the platform? So what you can do is you can sign up, you can go, and you basically just put your title of your experiment and why you did the experiment, and then you can drag and drop your files onto your web browser and upload them directly. And once you've done that, you have at your disposal a lot of information that you can just kind of deposit in. For example, on the left-hand side are your powerful actions once you've logged in. This includes your ability to make entire copies of experiments, which include not only the raw data, but the analyses as well. You can export statistics. We have an export statistics tool where you can export your results to an Excel spreadsheet. So you can, if you want to do further analysis, you can. You can also run SPADE, which I'll talk about in a little bit. You can also store any kind of metadata, which includes attachments. For example, if you know you did something weird with your protocol, you can upload your entire protocol, you can type it in, you can attach other things like images, spreadsheets, anything that is associated with your experiment that you don't want to lose. Finally, there's this part in the middle, which are your illustrations. Sorry, it's a little dark. But the illustrations is really the meat of where you do your analysis. This is where you lay out all of your figures, and you are able to share with your collaborators the different figures that you've made. Thanks, Wendy. So in addition, I wanted to highlight this part right here where you share with collaborators. So you can share an entire experiment with your collaborators. This means the gates, the figures, as well as your different kind of statistics and analyses. So how do I get to a figure in Cytobank, right? How does this work in terms of our platform? And so one of the things I want to show you, so this is an example figure that you can make within a couple minutes in Cytobank, and that's actually what we do in our trainings. Each row is an individual condition. So for example, the first row is unstimulated. Then you have IL-6, IL-10, and LPS stimulated. So pretty similar to what you're going to be doing this afternoon, tomorrow afternoon. Each individual column is a gated population. So your monocytes, B cells, and some of your different types of T cells. Then this is colored by median fold change against phosphoprotein stat 3. So everything's colored a fold change against the first row. So there's a lot of richness in this figure. And the question is, well, what if I want to start playing with it? What if I want to look at it in different ways? Each row in this case is a condition, and each column is a population. But you might have, during your data collection, maybe you ran more than just one sample, I would assume. And so really, on top of this, you might have individuals, for example. And in Cytobank, these are figure dimensions. And so when you're actually going through and doing your training tomorrow a little bit and analyzing the data, this is really key for how you set up your different plots. If you think about an individual sample, you might have 2 samples, right? You might have collected from 2 different individuals. You might have also stimulated with different conditions. You might have gated multiple different types of populations. And these are all different ways that you might want to look at your data and your experiment. And so instead of just one tube being an FCS file, it's really more than an FCS file. And the big thing, the take-home point from this, anything aside that I want you to take home today is when you're collecting your data, annotate. Annotate at the machine. Make sure your files have the names of your conditions and the different sample types that you have, because that is the most important thing that I can tell you today is to annotate your data. And so I think today you might be doing the LSR2 work, and Diva has this great way to kind of annotate after you've collected your data even before export. The reason for this that we take advantage of is that Cytobank, so on the right, your right-hand side here, we recognize a ton of things automatically. For example, the plate column, the plate row, and we bring that in automatically. On the left-hand side, for example, this is an experiment where I did 90 different therapeutics drugs. So you can see like rapamycin, things like that, over 8 doses, 2 cell lines, and in like 4 plates. And what you can see here is on the right-hand side, those were brought in automatically. I didn't have to annotate them. On the left-hand side, it was actually semi-automated, where you can basically just write that whole list in there, and Cytobank will find those files and annotate them for you. And so, for example, then each of these panels here is one of those figure dimensions I was talking about. For example, here are my conditions, here are my doses, here are my sample types. And you can collect a number of those as you're doing your experiment. Afterwards, you can create heat maps, you can create 2D plots. Once you have those annotations in, you can start stratifying and playing with your data in different ways. For example, you can look at individuals versus conditions for fluorescence data. You can quickly switch to histograms. You can switch to histograms in different ways and change how you're looking at them. And I just want to highlight this because you might be looking at this and think, well, this brings me to my original problem where I have a giant data set and I'm not sure exactly how to look at all of this information. And if you are someone who really loves to go through and do kind of traditional gating analysis, some of the things that we provide are gating strategies for CyTOF data. We put this gating strategy together with GVS. So this is an example on just a 17-color data set, which I know it's a lot of steps to gate all the way down to your different T cells and B cells. But what we're putting into the platform, and this is what we're actually having coming up in about the next month or so, is that this is actually the gating hierarchy for that tree I just showed you before, where you start with your ungated and then you gate down to your plasma B cells and such. And we are actually putting in Cytobank different visualizations so that you can interact with this in a more intuitive way. One of which is this sunburst visualization, which will be available in about the next month or so. In addition, we provide SPADE analysis for high-dimensional data. And so I'll talk about SPADE a little bit right now. But before that, I wanted to know if there's any questions before I move on. And I'll show you a little bit about how figure dimensions work in Cytobank. OK. That's either a good or bad thing.
So as you guys know, we'll be around in the afternoon today and tomorrow to help with all the analysis. So you can come and ask very specific questions during that time.
Yeah. So the other thing I want to show you briefly, I should have mirrored my screen. So these are those figure dimensions I was talking about. And you can actually select them, and you can search and look for certain ones. Like, for example, if I wanted P38, I can add that to my populations versus channels. And once I have those annotated, it makes it really easy for me to come down and look at my heat map and say, well, down here I can tell that I have certain channels, and I have these populations right here. But let's say I wanted to just pivot and look at that a different way. Well, I can just go up back to my figure dimensions, and each one of these you can actually drag and drop, such that now my channels are my columns and my populations are my rows. So I expect the transpose of this to now come up. And as you can see now that I have that exact same heat map but flipped. And this is something that you can export to a PDF to put into a PowerPoint slide, or you can even quickly change this. For example, if I wanted to change it to a histogram overlay, I can change it to a histogram and see what it looks like from that aspect to see how my data is changing. And so these are all things that you're going to learn a bit today and tomorrow while you're moving through and see how quick you can actually generate these types of plots. OK, so let me talk a little bit about SPADE. Black slides are not the best on this projector. So what is SPADE, right? So the general idea is that, so the original science paper that was published in 2011, it was at this point where they weren't sure how to gate it because, as I showed you, that gating strategy before was just kind of ridiculous. And you're collecting a ton of samples with very high depth. And you're not sure how to lay it all in one place, because if you go and you try to gate, you end up trying to backgate across like 15 different populations and moving back and forth within the data. And the way SPADE works is it actually goes through and it finds little clusters of cells in this high-dimensional space. So for example, if you're thinking, well, I'm interested in B cells, T cells, monocytes. And usually you go through and you dig through each of those individually. You'll go through and say, OK, I'm going to look at my CD19, CD20. I'm going to go with CD3, CD4, CD8. And I'm going to go like CD33, maybe my dendritic cells. What if I just said, I care about all of these markers? Please lay it out in a way that I can try to make sense of it within one universal map. That's what SPADE does for you, so that you don't have to kind of go back and forth while gating. You can kind of drill up and drill down in one place. And so what SPADE does, I think I might have removed that slide. Yep. sorry, is that at the end of the day, it generates a graph like this. So each individual node, I know it's a little hard to see, each individual node is a cluster of cells. And so usually when you gate, you're gating a population of cells. In this case, Spade goes through and says, okay, well in CD4, CD8, CD33 land, how many little populations of cells could I potentially have? In the beginning, you tell it. You say, I want like 300, I want 400 cell clusters. And I'll go through, and so each of these individual nodes here, each little circle is a group of cells. Each bubble is what you go through afterwards, and you say, well, I see that all of these nodes are CD3 high, so they must be my T cells. And you draw a bubble around that. Each color is the expression level of a marker. So for example, this tree right here, this spade tree, is colored by CD45. And then the size of each node is the number of cells. And the really useful thing about this is that when you build a spade tree, you're building it on all of your samples at the same time. And so what you can do is you can then do fold changes. For example, here on the left-hand side is your basal level for STAT5 signaling. On the right-hand side is the same kind of the original sample, but it's been treated with IL-7. Because you combine the data together and then you run Spade, it'll find those cells that are different in both. What you can see is faintly on the right-hand side right here, there's kind of this red population. And on the left-hand side, you don't necessarily see that right here in your T cells. However, what you can do is you can actually overlay the two trees on each other and get a fold change. And you can very clearly see that your T cells have a higher STAT5 signaling after treatment with IL-7. You can do this similarly with other signaling markers and look for specific signaling patterns as well. For example, down here, the satinib lights up this part of this tree here. One of the reasons why people really like this a lot in terms of analysis is because the steps to set it up are so simple. This is an example data set where, so Jonathan, one of our founders, collected 20 markers and basically went through a couple steps. Basically, you go through and you only gate maybe a couple populations prior to running Spade. In this case, he gated down to CD45 positive PBMCs, so only about 1 or 2 gates before moving forward. To set up the actual run, you only need 5 things. The first is the number of nodes starting out. In this case, it's the target number of clusters is about 400. The second is the downsampled events target. And so Spade goes through a couple different steps. The first is a density-dependent downsampling. So if you're interested in looking for example, rare populations, typically, the reason why we haven't replaced manual gating with completely automated tools is that rare populations are very hard to find. And so this downsampling step gives those populations a fighting chance of being found by the algorithm. So when you're actually going through and doing your analysis, you might be able to find some of those rare cells. Then you run Spade on a particular population. So typically, we'll go through and we'll do a cleanup gate, maybe a CD45 positive gate, and then run Spade. If you're interested in a very rare T cell subset, for example, then you might want to gate down to your T cells before running Spade. It's really a way to kind of universally orient your data and look at it and really drill down into some of those populations that you may not have known about before. It's really good for discovery of certain cell types. Then you'll choose your clustering channels. As I mentioned before, this is how you want to orient your data. The way you're going to be doing your analysis today and tomorrow is you're orienting it within space that you already know, meaning I'm interested in finding B cells, T cells, monocytes, so I'll choose those channels prior to clustering. The last one is Spade groups. This is where you'll choose your fold change prior to doing the analysis. All right, I'm going to skip these slides because they're dark. I'm just going to go into the actual website and show you. OK. So there's a couple different, so if you go to dvs.cytobank.org, we have a number of example spade trees that have already been annotated. And what you can actually do is you can clone and make a copy of that experiment, and you can rerun spade and do the analysis as well. It also gives you examples of how to analyze the data. If you go to, let me actually zoom out. So for example, right here at the top, as you can see, this is one of the examples that we were kind of showing before, where there's the target number of nodes, there's the downsample target, and then those three things that I talked about before. When you actually, after you've gone and run this, it will show this tree underneath. And typically what will happen is this tree is not annotated yet. It actually looks like there are no bubbles on there. And what you do is you go through, and Rob will talk about this a little bit tomorrow, but you can actually go through and you can switch through all your different markers. For example, whoops, I switch files. This is CD20, for example, so you'll see that my B cells light up. CD8, so my CD8 T cells light up. And you'll go through and you'll draw around a population and label it. I'll show you an example here. So for example, here are my CD8 T cells. You can actually go through, you can select a subset of nodes, you can move them around, and you can also color by a certain marker. So for example, if I color by CD20, I can definitely see that some of my B cells light up. Try to pick another good marker to look at in this case. CD3, for example, this whole bottom section lights up because you have your T cells. CD4. And you can basically go ahead, you can select a set of nodes, and then you can label them and orient them. And so that's, for the original science paper, if you take a look at it, that's how that original spade tree was generated, where you really get to play with the data all in one place, and you don't have to kind of go up and down while gating and look at it. The other benefits of this is that to really trust how you're doing in terms of your analysis, you can actually pop out a number of these plots, which are these 2D plots, for example. Let me pick, so for example, this is CD20 versus CD8. And if I choose, for example, my CD8 positive T cells by themselves, you can see that they're CD8 positive, but they have no CD20. And that way you can really decide those cutoffs for your analysis. And you'll definitely see some trees in some of the presentations today and tomorrow. And you'll notice that all of them kind of have these different shapes in terms of how you can analyze it, because you can actually go through and you can really work with the different cell clusters and orient them in a way that makes sense. Any questions so far about that? Yeah.
I'm kind of new at this, but it kind of
Sure.
bothered me that you could move a population, because I thought it meant something where it was
Yeah.
located.
So you can think of this kind of as, like, you know, let's think about the map of the globe, right? And basically, so each edge, the actual distance of the edge doesn't really mean anything in this case. But the fact that these are connected to each other does mean something. That means that they're close to each other. If you take a map of the globe, for example, and then you lay it flat on the page, you notice that everything kind of gets moved a little bit. Like Greenland is huge, right? But things like Alaska and Russia may be very far apart from each other now, but at least you know that Alaska is close to Canada and everything like that. And so the spatial orientation, you do lose a little bit of information because you're taking something that's like 25 dimensions and putting it down into 2. But at the end of the day, you do have a lot of those kind of orientations. Yeah, but it's true. Anything.
So then how was the initial layout?
Yeah, so the initial layout, let me actually show you an example. So this one, the one I showed you the gating strategy for, this is the spade analysis for it. And if I go back, we actually showed tiers. There's 4 iterations for how we did the bubbling. Oh, I don't think I have the original tree. But the original tree is actually just laid out relatively randomly. And so basically, after it's found all of those kind of elbows and knees, I guess, for the entire tree, it just lays it flat on a page. Here, I can show you an example of one. Let me just clone this one real quick. So what you can also do is, if you have a particular tree that you've done, you can actually clone I don't have access to this one. Let me just clone this one.
So essentially, you decide on a layout that you'd like, and then you can clone that layout for your different experiments.
Yeah. But naturally, a lot of that, so if you look at the original science paper, it has the hematopoietic lineage in it. And so naturally, a lot of those things that are close together has a lot of biological information within it. You just have to lay it out within the way that you understand the way you've chosen to begin with. So let me reset this visualization and show you kind of the naked original tree and what it looks like. When you do your analysis for, so it looks like that. And there's a lot of places that you can kind of tweak and play with how things look. And what you can see is that there's always a guide for how many cells are in a node and the color scheme, et cetera. You can actually change the color schemes as well. Okay. But yeah, it goes back to that whole thing I was talking about before, where everything's linked down to the raw data. And so what's really beautiful about this is that what you can go is, once you've bubbled these populations, you can actually export them to a new experiment and create your heat maps, create your histograms, and do all the analyses that I mentioned before. And let us know if you have any questions about that as well. Okay. Once you've exported to a new experiment, and you can also do this in your original experiment, we actually have our export statistics tool. So medians, coefficient of variation, standard deviation, variance, a lot of statistics that you might want from your data, you can actually export to an Excel spreadsheet. And then I just showed you some of those example trees. Yeah, that's a starting tree. And then basically, you're kind of going through and you're going through the analysis how you would, similar to gating, but you're doing it all in one place. And that's what allows you to kind of go through and find those populations of interest. Okay. For those of you, so you're going to have some training tomorrow. And then kind of moving forward, if you want extra training, we actually have a number of data sets that we do either webinars or one-on-ones with. The one that you're going to be doing is very similar to this one, where you have your PBMCs and you've stimulated them, and then you collect some core panel. And then afterwards, you're going to be able to explore it and see what the results look like in terms of which populations are stimulated, how do they look. We typically do our trainings on the PBMC data set, but we also use the original science paper data set for training for people as well, where we show them how to do not only the heat maps and histogram overlays, but also spade trees and export of statistics. I just wanted to kind of take the last 10, 15 minutes to talk about the other resources available to you, a lot of which are just available online for you to take a look at. But prefacing that, just to give you an idea of the platform as a whole, here at Stanford, one of the things that we do is that the Stanford Flow Core facility actually stores all of the data that they collect, and they actually have a Send to Cytobank button where a user can immediately upload to Cytobank directly from the core facility. In addition, sorry, I'm losing my voice. We have other resources available. For example, we have our plate annotator. So that data set that I showed you earlier where I had 90 conditions, if I had made a mistake or if I wanted to add extra annotations to the columns or rows of my plate, we actually have a mass plate annotator where you can go and analyze your data. We also have a dose response analysis part of Cytobank where we really appreciate feedback as we work on improving it and moving forward with it, where you not only generate the log EC50 curves, but you can actually mouse over and see the underlying original data with it, which is a theme throughout Cytobank. In addition, I mentioned that we have SPADE available and that we're coming out with other visualizations and techniques for analyzing high-dimensional data. What I didn't talk about was kind of collaborative sharing through Cytobank. Not only can you share your full experiment with your collaborators, but you can work in teams using projects. So if you look at cytobank.org, which is our free public version, you'll see over at least 80 data sets, maybe more, that are just made publicly available by people, because what happens is that we are a really easy place to put the data up when you publish a paper. And so you can actually make a data set public when you want it to be public for everyone to see. They can't go and edit your original analysis, but they can go and they can see the figures that you did for your paper. In addition, we have projects where you have project managers, you have project members, and they have different granularities of sharing. Like if you made a project and you didn't want someone to edit all the gates, you can do it that way, or you can share just the figures as well. And so that's something else that we have within Cytobank for sharing as well. The last thing I want to mention is if you have signed up on Cytobank before, for example, cytobank.org, and you wanted to try running Spade, for example, or you wanted to do some kind of advanced plate annotation, as far as I know, we're the first people to implement the ACS container, where you can actually take your entire experiment from one Cytobank, package it up, and move it to another one. And we can do that for you, or you can actually do it yourself. If you wanted to move one whole experiment with your gates, with your analysis to another one. And this is really useful for collaboration. What we've noticed, especially for some of our European customers, they've been actually collaborating between sites that are hours away from each other. They've been moving between Cytobanks for analysis in teams. The last thing, which I think you all probably have in your notes, but you probably want to take notes on in general, are all of our community resources, because we put a lot of effort into kind of freely available resources for people to use. One of which is the Nolan Lab signaling resource, which is available at cytobank.org/NolanLab. So the stuff that you're doing today, the protocols that you're getting, those actually are available also online under experiment protocols on the website. There's also conjugation protocols as well as Cytobank reports. And so the Nolan Lab, as well as Bodemiller Lab now, who used to be here, basically what you can do is prior to publication, you submit, you can put a lot of different, your figures, your supplementary info all up on Cytobank, and we can create a report for you. And so when it's published, you can simply point the reviewers prior to as well as after for any people coming to the paper to the tons of data that you have as well as the analysis figures. I'll show you that more in just a second, but just to give you an idea, example Cytobank reports includes that breath experiment that I talked about where we had a ton of kinase inhibitors from 2012, the viability staining paper, as well as the bead-based normalization paper and the cell cycle analysis. The most recent report that we generated was for the Bodemiller Lab, which this is their new landing page through Cytobank, and they actually did imaging data, and then they took the individual, so they did mass cytometry-based imaging, and they took the actual information and put it into Spade. And so you can actually go and you can access and view the data and even download it through Cytobank. Let me just show you real quick what the Nolan Lab resource has. Somehow that got smaller. So what you can see here is that there's the page that I was mentioning before, and then you can go to experiment protocols, and you can also go to reports. For example, Rachel's paper, the normalization of mass cytometry data, you can actually go and you can download the software for doing bead-based normalization, which you'll be doing tomorrow as well. And then there's a number of figures that are all generated on Cytobank, which you can actually go back and you can view the raw data in Cytobank as well. And so similarly to when you do a publication and there are versions, we have the same platform where you can actually have a version of a figure post-publication that syncs up for the that can be referred to within the publication itself. Okay. The other thing I wanted to talk about, actually I'm just going to show you the websites instead of going through the slides. So DVS Cytobank, which I mentioned a little bit earlier, we're in the middle of rebranding it to Fluidigm Cytobank. But there's this nice link down here called Get Mass Cytometry Data on the left-hand side, where you can actually download the protocol sheets as well as information about the panel kits that they have. So that gating strategy that I just showed you, that giant gating strategy, we actually put that together under the human PBMC kit. And we're generating those for T cell analysis as well as like mouse splenocyte analysis as well. The other resource that I wanted to show you is BD Fact Select, which is this resource here that we have together with BD. If you are interested, for example, in measuring ERK, and you click on this, there's underneath, there's the different types of buffers that they have available, and you can see as you use them how good your signal difference is within these different permeabilization buffers at different concentrations. And you can view the original data in Cytobank as well. And so that's really good if you're trying to look at different antibodies and buffer combinations and thinking about whether or not you're going to get the signal that you need. And so that's, yeah, it's gotten a lot bigger recently in terms of if you pick your favorite signaling or surface marker, it should be on there. Okay, let me see what else I have on here. So other resources, we have our file concatenation tool, which is available through our support portal, and that's the last thing I'm going to talk about. When you're collecting data off the CyTOF, you often have a lot of little files, and you might want to concatenate them. And so we actually have a concatenation tool on our support website that you can get access to. You can just download it for free. We make it freely available to the public, which I will show you our support website now. So when you leave here, one of the big things that we really want to help people with is getting up and running on their analysis and getting their results quickly. And so if you go to support.cytobank.org, we have our blog, which every month or two we feature a new researcher and the kind of work that they're doing with Cytobank. But you can also get a lot of support within, usually within a day or two from us. You can email helpdesk@Cytobank. We also have our chat room. So if I go directly to the chat room itself, there's always someone in there who can respond quickly. If you ever do go to our chat room, please write your full name so we can actually email you afterwards if we miss you. Because often someone will come in and their name's like John, and there's no last name. And we're just really sad because you're trying to figure out what they wanted, and you have no idea. And so this is the chat room here, and you can type and say hello. Whoops. But the nice thing is that our operation director, he has 2 young children who are about 2 years in age. So even if it's 2 a.m., I've gotten a text at 3 a.m. saying someone needs help in support chat. And so please don't abuse that power, but just to let you know that usually there's someone in there. And at the very least, we'll email you afterwards. We have a number of video tutorials. We have our YouTube channel where you can get started on just basic Cytobank as well as these different things I've talked about today. With figure dimensions, compensation, we have 4 different videos on compensation, as well as other types of analyses. And we have our blog. And so just to take home at the end of the day, it's all these things I talked about where we really have a huge emphasis on education, but we also really care about the managing and export and sharing of your data, as well as kind of that preventing loss and allowing communication to go from person to person as new people come through the lab, right? And keeping that going. What did I measure in that experiment from 6 months ago? Think about years ahead or behind, and that's really where we try to fill that niche and that need. In addition, right now we have a number of security certifications that are coming out. We're working on a white paper if you have questions about that. Any experiment that you put on Cytobank, it is personal to yourself, and you have to share it with other people. And so you don't have to worry about other people suddenly adding themselves to your experiment. It is by default private to yourself. These are the resources I mentioned earlier, and I think they should be in your slides as well. So you're welcome to contact us with any questions and comments. Nikesh and I will be here today, tomorrow, and in addition, we're available by email as well. Thank you. Any questions, comments? You you won't learn until you actually do. So I tried not to go into too much of that detail, but it'll be good. Cool. All right. I'm the only thing between you and lunch, so I'm going to let you go. Thanks for listening.